crop plants may be contaminated with viruses. These viruses pose a threat to the success of various vegetable or ornamental crops around the world. In general, viral diseases will lead to a loss of vigour and a decrease in the quality of fruits or flowers. When a plant is infected with a virus, as plant does not possess a true immune system, as in humans or animals, and because there are no adequate plant protection treatments, the virused plant is condemned to remain sick all its life. For crops grown from seeds, the problem is less crucial because sexual reproduction could, in some cases, eliminate viruses. On the other hand, for crop plants that are multiplied by vegetative propagation, to obtain uncontaminated material, there is a laboratory technique that allows the regeneration of healthy individuals. This technique consists of meristem cultures. It may or may not be associated with thermotherapy. Meristems are small niches of pluripotent stem cells that are found within the buds and ensure the differentiation of organs. These meristems are the only tissues originated from an infected plant that can be free from viruses. When these meristems are in vitro cultured on a culture medium and under controlled conditions, these meristems can regenerate plants that are free from viruses. Carnation flower is one of the well-known ornamental plants found in most florists and is produced all over the world, but as many plants, it is sensitive to many viruses. The culture of meristems can be used in order to produce healthy material. It is this flowering plant that constitutes our study material for practical work. Arabidopsis halleri, or Halleris arabette, is a wild plant studied in our university. Its interest for scientists is based on its ability to grow on soil contaminated with heavy metals. For research purposes, Arabette plants are grown in greenhouses and vegetatively propagated. It is during this stage that the viruses could be transmitted from plant to plant.
We're working on Arabidopsis halleri, which belongs to the Brassicaceae botanical family. This plant model exhibits two main characteristics. The first is that it's genetically close to Arabidopsis thaliana, a well-known plant model in genetics. It led us to perform molecular biology on this species by adapting the tools previously developed on Arabidopsis thaliana. The second is that this species is considered as a metallophyte plant that could grow on polluted soils, especially soils polluted by heavy metals. We're investigating a physiological competence called metal tolerance, that is the ability to develop on such degraded soils. First, we have to collect plant samples on polluted and non-polluted sites for comparison. From these two populations of plants, either we perform genetic crosses to establish how this tolerance character is segregated in offspring between individuals that grew on polluted soils and individuals that grew on non-polluted ones, or random samples are taken among these two populations. Then the level of metal tolerance is determined according to the populations. Experiments were achieved under controlled conditions in growth chambers. We have performed several waves of prospecting in Europe, as in Germany, Poland or Italy. During the last collection of samples, we went to the Italian Alps, and we brought back from this region individuals of polluted and non-polluted sites. A few days after their introduction into the greenhouse, unexpected symptoms that had never been observed on our plants appear as brown spots surrounded by a light background, or inversely, we don't know how it appears exactly. In fact, it looks like a mosaic phenotype with dark areas and clear areas. These colors, or symptoms, indicate clearly the appearance of a virus disease whose presence has been confirmed by the phytopathology tests such as ELISA and observations by electronic microscopy. This viral attack impacts the health of our plants. This viral disease impacts significantly the vigor of the plant, comparing to those previously collected that were still pretty and well developed. Such plants could easily be propagated through vegetative cuttings. Usually, these plants could naturally cut themselves through the production of stolons. After a few days of viral infection, plants appear small and all stunted and exhibit small leaves. As a consequence, self-cutting seemed no longer possible. Indeed, all cuttings died, and no more experimentations could be undertaken if no plant material was available in quantity. It seems therefore necessary to eliminate this virus. This is not easy, as all of our plants are concerned. The experimental strategy we've adopted is to try to produce plant cuttings from any intact parts, even small, and especially the virus-free parts. Meristems could help us to reach this goal.
In order to cultivate meristems, the plant material must be disinfected before. This constitutes the first stage of culture. Carnation exhibits a stem with opposite decussated leaves. It means that the pairs of successive leaf insertion points intersect at right angles. Leaves are inserted on bulged nodes. The ornamental flower is found at its apical end. In the axil of each node, we find the axillary buds, in which the meristems are found to be further cultured. The dissection of a bud is performed after the disinfection of the plant material. First, stems should be cut into nodal fragments that will be disinfected. For sampling, the fragments are collected from the bottom to the top of the stem. Collect is stopped at 2 or 3 nodes above the flower, as the last meristems are usually converted into flower ones. For the Arabette, the development of plants is different from the carnation's one. The Arabette is a rosette plant. It is therefore the apical meristem that will be removed and put into culture. The Arabette tends to multiply naturally from the axillary buds, so we will find on an adult plant several shoots that will be separated from each other for disinfection. They are called cuttings. When all nodal fragments are excised, from carnation and Arabette, the disinfection is carried out under a laminar airflow. The plant material is placed in a flask previously sterilized in an autoclave. For the carnation, the fragments are first immersed for 2 minutes in a 10% mercuril bath in order to permeabilize the tissues of the carnation to promote disinfection. The mercuril is then removed. And a rapid washing is carried out with 70% ethanol. Then the ethanol is replaced with a solution of calcium hypochlorite at 70 grams per litre for 10 minutes. After this, the disinfecting solution is removed. and 3 rinses are performed with sterile water of 5, 5 and 10 minutes. For the Arabette, the nodal fragments are immersed for 20 minutes in a solution of calcium hypochlorite at 80 grams per litre, supplemented with a drop of mercurol. Three rinses with water are then carried out as for the carnation. The material can then be dissected under aseptic conditions for the culture of the meristems. For the dissection, leaves that mask the axillary bud were removed. As the leaves are less sheathing, it's necessary to carefully remove the leaf to the base. This allows the axillary bud to be released. A binocular microscope is necessary to perform dissection under a laminar flow hood. The bud is made up of foliar leaves that protect the meristem. They are smaller and smaller as you move inwards. In order to reach the meristem, we will therefore remove the leaf primordia one by one, beginning with the most external ones. They are generally arranged 2 by 2 as the mature leaves. Leaves and leaf primordia are removed up to the base of the bud. The lead primordia pair is arranged in the opposite direction at right angles to the first pair. As leaf pair that covers the rest of the bud is removed, the following foliar pair appears. In some cases the meristem may already be visible, or it's just necessary to remove the third pair of leaf primordia, which are arranged in the same way as the first pair. The leaf primordia are gently removed by spreading on the side. The leaf is lifted and the meristem appears, which resembles a small dome, the appearance of which is more brilliant than the foliar primordia. Some very small leaf primordia could be taken at the same time when the meristem is cut. To remove the meristem, a scalpel is used. Cut to the base. And the explant is based on a solid development medium in a Petri dish. The Petri dishes are closed with parafilm and then placed in the growing room. leaf formation will occur at the periphery of the meristem. A small stem will then develop between each pair of leaves. When a few leaves have developed, the main root is initiated at the base of the leafy stem. Lateral roots then form. When the small shoot has reached 1 to 3 centimetres, it can be transferred to a development medium. The transfer takes place in culture tubes. Until the seedling reaches about 10 centimetres. After the surface disinfection, in vitro shoots from Arabette will be dissected by removing one by one the leaves that surround the apical bud. When the leaves become too small, the binocular lens is necessary for achieving the dissection. The leaves are removed to the base of the petioles to clear the apical bud. The 3 to 4 pairs of leaf primordia are progressively removed. To remove the meristem, a scalpel is used. Cut to the base. The meristem will be cultured with 2 pairs of leaf primordia in small petri dishes on solid development medium and then grown in a culture chamber. Small leaves will grow in rosette around the meristem. A root will develop later. When the seedling has reached a size of about 2 centimetres, it will be transferred into a plastic square box called a magenta box for further development. When the plants have grown sufficiently, they can be transferred to a greenhouse for later use. The moment when in vitro plantlets are transferred to soil under greenhouse conditions is called the acclimatization phase. It requires some precautions. For this purpose, the plants are removed from their culture vessel and washed in order to remove the agar medium, which remains stuck to the roots and which is capable of promoting the development of fungi. They are transferred into pots that contain a lightweight substrate, such as peat, that will allow good root development, and in mini greenhouses in particular to avoid desiccation. Aeration may be provided to prevent fungal contamination. The transition to a less humid atmosphere will be done gradually in order to obtain mature plants completely acclimatized. The culture of carnation meristems has been used for a long time to suppress viruses. Numerous papers establish the elimination of viral diseases and a reversion to a healthy status. For Arabette plants, there is currently no evidence available to prove that the virus has been eliminated. Plants originated from such in vitro process are no longer showing mosaic symptoms on their foliage and have significantly improved development compared to the initial plants that were contaminated. Virus assay tests exist and can be used to determine the health status of such plants. The cultivation of meristem is therefore a technique of choice, which has allowed and which always allows to save many plant species and cultivated varieties.